Today, we'll talk about post-GWAS analysis. So in this lecture, we will talk about heritability, the LD score regression, the genetic correlation, and annotating GWAS results. So first, let's talk about heritability. What is heritability? How to estimate heritability, and how to explain the missing heritability. What is heritability? Heritability is simply the proportion of phenotypic variance explained by the genetic effects. There are two major definitions of heritability. The first one is the narrow sense heritability, usually annotated by small h squared. The narrow sense heritability measures the proportion of phenotypic variance explained by additive genetic effects. It ignores the interaction or other non-additive genetic effects. The broad sense heritability broadly measures all the genetic effects, including additive and/or non-additive genetic effects. And the broad sense heritability is typically noted with big H squared. So, how to estimate heritability? There are three ways to measure heritability. In the past, before genomic technology enables the assay of large-scale genomic variance in the genome, people use family data to estimate the heritability. This estimate is able to estimate the proportion of variance attributed to all genetic factors, including all genetic variations across the frequency spectrum. So let's take a look into how this is done. It's actually not very hard. It can be done by comparing the concordance of the phenotype measurements in monozygote twins and heterozygote twins. We know that in monozygote twins, the genetics are exactly the same across the twins, so you have the full contribution from the genetic component plus the shared environment of the twins. For heterozygous twins, we know they only share half of the genome, so the contribution from the genetics should be divided by half because only half is shared, and also there is the same shared environment because they live in the same household. So what you can do is you abstract the concordance between the monozygote twins and the heterozygote twins, and you time this number by 2, that's going to be the measurement of the heritability. As simple as that. So here is an exercise. If you have the concordance of phenotype for monozygote twins as 0.8 and for heterozygote twins as 0.5, what is the heritability? Here is the answer. You simply subtract 0.5 from 0.8 and you multiply this number by 2, and the answer should be 0.6. Luckily, to date, the heritability based on twin study has been beautifully compiled in a website called Match. You may simply go to this website, pick your favorite trait or disease of interest, and you can take a look at the heritability from these estimates, from the twin and from other methods in estimating the heritability. You don't really need to do it yourself because people have done it for you. The second method for estimating the heritability is to use genome-wide significant SNPs. Naturally, as you all have learned how to do GWAS, it's natural to use the significant SNPs from the GWAS as they represent the true positive findings of the association between the SNP and the trait. It's natural to use them to calculate the heritability. To do this, this is very simple. You first need to identify which SNPs are significantly associated. You can conveniently get them from the GWAS result. You estimate for each of them how much phenotypic variance can be explained, and you sum them together. And this is covered by a paper published in 2012 in Human Genetics. However, you will notice that, interestingly, the number estimated using this method, using the genome-wide significant SNPs, is much lower than the number you get from the twins. For example, in Alzheimer's disease, the number of the heritability estimate from the twins is high as for example, for the Alzheimer's disease, the heritability estimate from the twins is as high as 0.79. But if you only take into the significant SNP from the GWAS, you will be able to explain only 18% of the heritability. So that's a big difference. And the same thing applies to all other disorders, including bipolar, breast cancer, cardiovascular disorders, so on and so forth. The phenotypic variance explained by the significant SNPs is just a proportion of the heritability you were able to find using the twin studies. And this is covered very beautifully in a Nature article published in 2008 and termed as the missing heritability. This missing heritability actually haunted the field of genetics for quite a few years. There are many explanations, and I have to say that there's still an ongoing debate which of the and I have to say that it's still an ongoing debate which of this explanation can be and I have to say that it is still an ongoing debate which, or a combination of which explanations can be used to explain the missing heritability. So the explanations can be the heritability estimated from the twins can be overestimated. It can be that the GWAS is not well powered to detect the rare variants because, as we know, the GWAS mainly targets the common variants, and there is a big proportion of the genome as rare variants which are not being studied by the GWAS. And there could be population-specific variants, meaning that if we're only using the European GWAS, we are not able to find the heritability explained by variants enriched in the African or in the East Asian population. And another explanation would be the phenotypic character. And another explanation could be the phenotypic characterization. We know some diseases can be very heterogeneous. If you were only able to ascertain some subtype of the disease, not the other subtypes, you are not able to capture the full set of the genetic variants associated with all the disease subtypes. And the other explanation would be the common variants that have very small effects because the effects are very small. Even if we have thousands or tens of thousands individuals, we are not able to pick them up. But the last hypothesis can actually be tested as we now have technology to use the genome-wide distribution of the test statistics for common variants to infer how much can be explained by the common variants that are not significant yet. And here we're looking into how. So this technology is able to include all the common SNPs on the genotyping chip and to use the distribution of the test statistics of these SNPs to calculate what the maximum of heritability can be explained using these common SNPs. There are two methods can be used for this purpose. One method was published by Dr. Peter Vicious' group in 2011 in American Journal of Human Genetics. This method requires the individual level genotype data and is able to give a very accurate estimate of the heritability explained by common SNPs. The other method was published more recently by Dr. Ben Neal's group in 2015. This method only requires the summary statistics instead of the individual level genotypes. However, the estimate from this method is less accurate comparing with the GCTA, which is the first method. So, what if we use one of the two approaches to look at our data? We take height, for example. If we use a twin study, we get the narrow sense heritability as high as 0.8. If we only use the SNPs that are genome-wide significant, we get the heritability explained by these SNPs as low as 0.1. If we use GCTA to capture all the common variants, we get a heritability estimate of 0.45. This is a big improvement from the 0.1, but still it doesn't explain all the heritabilities estimated in the twin study. The explanation could be, as we said, the population-specific variants or the rare variants. So here is a figure summarizing all the heritabilities we have talked about. We have the heritability estimated from the family study as the highest heritability captures everything. And we have the heritability characterized by the genome-wide significant SNP in GWAS, and we have heritability estimated using all the common SNPs, which is the value in between of them. And as the sample size increases, we will be able to find more SNPs significantly associated with the trait. Therefore, the heritability explained by the significant SNP will increasing and will eventually approach the heritability explained by all the common variants, assuming we have large enough samples. So, in summary, we introduced the concept of heritability and how to calculate heritability. To calculate heritability from all common SNPs, we may use GCTA and LD score regression. GCTA requires individual-level genotype and is very computation intensive, but it gives you a more accurate result. LD score regression, however, doesn't require the individual-level genotype and is therefore less computation intensive, but it gives you less accurate result.